Hi, I am Madhurima Datta, Zoology and Biology Educational Content Creators. Now's content for explanation of research paper of zoology or biological sciences. Paper non-coordinates and topic is developmental of basal bodies in Paramecium. Paper of by Ruth V. Dippel, Department of Zoology, Indiana University and Bloomington, communicated by T. M. Sonborn. It is published on August 2, 1968. So, introduction. Basal bodies or centrioles occurs in the cytoplasm of almost all animals and lower plants and are implicated in the formation of flagella, cilia, and certain sensory structure. It is the most important structure in mitotic apparatus. Previous researchers have been reported that for early developmental stages have observed at the site where the new structure will develop either a sphere of dense amorphous material or a young organelle shorter than the adult. But nevertheless, processing most of all of the features characterizing the mature pattern. In early development stages, kinetosome, that is ciliary basal body of paramecium, a mode of development that may be expected also to apply to basal bodies and centriole of other organisms. Methods. Paramecium aurelia stock 51 was grown at maximal rate in the wet lattice medium 2 and 270 degrees Celsius. Success in detecting the earliest stage of basal body in formation was based microliter on recognition of the three facts. First, the new ciliary basal body arises at right angles and in closed approximately to the anterior portion of an existing basal body. Second, basal body proliferation occurs first and mainly in a zone encompassing that the middle third of the body. Third, maximal basal body production is reached within a 20 minutes period, beginning approximately 50 minutes prior to the completion of cell division. They reported at this time the cell has begun to broaden anteriorly but as yet showed no sign of a cleavage furrow. Single cells at this stage were fixed by them. The 1.5% veranal acetate buffered butyraldehyde fixed in 1% 7 buffered osmium dehydrated and oriented in upon to yield both longitudinal and the cross section through the developing kinetosomes. Section were stained with uranyl acetate and lead citrate and observed with Hitachi heat. Next, the mature organelle, like the mammalian centriole and the ciliary and the flagellal basal bodies of the other eukaryotes. The basic architecture of the paramecium basal body consists of a cylinder whose wall is composed of 27 microtubules arranged in 9 equally spaced groups of 3. The 9 triplet sets run parallel to the long axis of the basal body, but each set is pitched at an approximate 40 degrees angle to the cylinder surface. The outermost tubule of each triplet is designated as C and the innermost is A. The individual tubules are approximately 200 to 230 Armstrong in diameter, have a globular subunit structure comparable to the tubule composition. Next, A and B of each triplet continue into the cilium to form one of its 9 doublet sets, but C terminates at the basal plates. In the proximal portion of the basal body, the triplets assume a more acute angle with respect to the cylinder surface and connections become evident between the A tubule of the one triplet and C tubule nearest to it in the adjacent triplet. The complex of half 9 spokes and the 9 triplet tubules is commonly called a cartwheel. In Paramecium, the half frequently appears biotripartite at the spokes segmented and interconnected by fibers. Basal bodies of protozoa frequently also bear various appendages. In Paramecium, to serve as important landmark to pinpoint the site of the basal body origin, a broad kinetodesmal fibers whose root derived from the 3 anteriorite triplets and a linear set of 4 or 5 laterally directed tubules. and originating across the anterior base of the basal body and sloping upward toward the animal's left side. Next, basal body development. Stage 1, the generative disc. Basal body synthesis starts with the appearance of a flat disc of dense fibrous material near the left side of the kinetodesmal fiber and immediately anterior to an adult basal body and its set of lateral tubules. The disc is circular in outline, slightly smaller than the mature basal body, and shows no internal organization. A distance of several hundred angstrom intervenes between it and the adult basal body. Serial sections of this region were examined particularly for connections between the old and new structures. Next, although there are no microtubular connections were found, the 9 fibrous materials surrounding the triplets appeared confluent with flat of the disc. Next, stage 2, formation of a ring of singlets. Looking anteriorly and viewing the following developmental stages from the posterior side of the disc, one observes the microtubule assembly always begins in the upper right quadrant adjacent to the kinetodesmal fibers. Individual tubules then develop consecutive at equally spaced intervals of approximately 400 around the disc rim until a circle of 9 tubules is completed. Seen from the posterior surface, the first few additions occur counterclockwise to the original tubular, but later stages suggest that they may also form in a clockwise direction. Conceivably, the spacing in the accomplished by short fibers or connectives observed between adjacent tubules in the developing ring, a suggestion consistent with the fact that once the ring is established, the connectives disappears. These tubules constitute that the inner or eight tubules of the 9 triplet cells in adult basal body. Addition of disc material appears to accompany this stage. Next, stage 3, formation of double sets. When 6 to 7 single tubules are present, a second round of tubule proliferation is initiated, again in the upper right quadrant. This round may not always be sequential. B tubules develops as an outgrowth from a single site on the first, starting as a short stub which elongates arcs into a C and establish contact at a second site on the singlet approximately 900 counterclockwise from its point of origin. The B tubules is therefore an incomplete structure sharing a quarter of its boundary with A. Next, as the last stage, stage 4 is triplet formation. A third round of tubule production is initiated before completion of the second and follow a similar development pattern with the third or C set of microtubules growing from and to corresponding sites on the tubules of second ring. At the same time, the new basal bodies begins to elongate rapidly, a fact probably accounting for report that in adult basal bodies and centrioles, the C tubules sometimes appears to be shorter and its proximal end than the other two tubules of a triplet than the other two tubules of a triplet. Slightly later, a fully organized hub and spoke structure is recognizable in the young basal bodies, which by now is approximately 100 meter per Armstrong long, but still oriented at right along to the old one. It differs from a mature basal body, however, in the steeper pitch of its triplet, which still lack the A to C linkers, internal helix, organized central mass, vesicular and fibrous components associated with the cartwheel and the accessory outer appendages. Now the today's topic has ended.